Okay. One thing is whenever there's, you probably already know, that when there's a whole bunch of slides, I have to go through and erase each individual single one. Otherwise it does that in a little automatic space, so you can just like scroll through the page you have to go through till you hear something. Okay. But yeah, so once again, this sort of thing, for supercoiling, when you run it out in a gel, usually you get multiple bands or smeared because it can actually have different shapes on the gel electrophoresis. You can imagine it being supercoiled, being wadded, a little ball being looking more like a slinky kind of thing. If you cut both strands, it's going to be linear. Okay. So now we're going to really get into the nuts and bolts. Remember, this is not on tonight's exam. It almost made it to tonight's exam, and I remember we hadn't discussed it yet. So if but all of chapter 8 will be privy, I guess you could say, for the next exam, not tonight's class, and the final as well. So in eukaryotes, the packaging is much more complex, and there is a lot of research on this. Let me make it a little smaller so we can see the actual numbers. I also would show this in forensic science, talk about the beauty of it, and I don't know, non-majors, they could care less, I think sometimes. But there's the DNA double helix with the relative size. Don't memorize the size. It's about 20 angstroms, give or take, in width. Then the first in the hierarchy of DNA condensation, the first step is the idea that they start to form nucleosomes. Nucleosomes are composed of histone proteins that get the DNA wrapped around it. Kind of reminds me of like a spool of thread. It gets wrapped around it. We're going to go into a lot more detail of histones in a moment. Okay, histones, the way they're numbered, they're given like a letter H and then a number. For whatever reason, historically, they have weird numbering. It doesn't make sense. They're not numbered 1, 2, 3, 4. It's like 1, 2, 3, B, 3, 8. And it's kind of, I don't know, historically, I don't know why. Then what happens is the individual nucleosomes themselves start to accumulate. They get attracted to each other as well. And so that condenses further down to make chromatin fiber. And now we've gone from a single DNA being 20 angstroms to now we have a lot of DNA. It's only 300 angstroms, okay, for the diameter. Then we have the big, and to me it kind of looked like if you ever went to the grocery store and you looked at garlic, you know how they put the garlic on the little bunches? That's what it kind of reminded me of. Or I guess you can also think about those, like those really, to me it was cheap, like old lady beads, where there's a bunch of little beads on the necklace. But then you can imagine the clothes of garlic and beads or whatever, that they can start to collapse and come down to each other. And so it helps fold even more DNA and to wrap around. And so as that starts to wrap around, during metaphase, you get the actual chromosome. And so the chromatids themselves are 1.4 microns, about 1,400 nanometers. So that's about 0.1 nanometer. Oh, I can't do math in my head. But we've gone through many several thousand-fold condensation of the event to make the actual DNA. So we have a relatively small space to make a chromosome. We've got lots and lots and lots, billions upon billions of base pairs of actual DNA. So we're going to talk a lot about the histones themselves. Histones are proteins, but they are highly, highly, highly modified. There's a lot of research on this. In fact, this is just a diagram here of some histones. So you see 2A, 2B, 3, and 4. Don't ask me where 1 is. Okay. So histones in general, do you suppose are they going to tend to be positive charged, negatively charged, or neutral? Why would they be positively charged? Because they bind DNA, DNA is. And why is DNA negatively charged? Phosphorodiazed back. Okay, so now a lot of that, we have to remember that with respect to all these different modifications. And now that's why you need to be able to use the logic based here in order to explain the ramifications and such. And so these histones The way that this is taken from a Nature Review of Cancer, but I don't remember exactly which edition. This is fairly old. But we can see different types of modifications on each of these. Histones are rich in amino acids. Like if it's going to be positively charged amino acids, what are some of them that can be positively charged? Lysine is the biggie, as you can see here. All the K's, it's one of the ones that are code. But they also many times will have like serines and things like that, which can do polar bonds and hydrogen bonding. Okay. All of these can be modified as well. Okay, so we have, this is the N terminus, that's the C terminus for H2B. That is not an asparagine, and that is not cysteine. That means N and C terminus. These numbers refer to the human location, I do believe, if I remember correctly. Okay, and then these are the modifications that can occur. So we have an A, we'll talk about what the A means. We have a UB, we have a P, you have an M. I don't have a simulation on this one. But we'll talk about that in a moment. So that's what this was showing here. Is we're looking at this point, and that's just to show that the C terminus, or near the C terminus here on these, they can be ubiquitinated to help decrease their affinity for DNA. So if you know, that's what the A stands for. All these A's stand for acetylation. And so we have lysines. Lysine, what functional group is at the end of a lysine? An amino group. Okay, now let's fast forward once again. I did have it right there again. Okay. So this is another one. I don't remember what paper I took this one from. It goes into more detail. So once again, they've got the numbering for the various lysines primarily. We believe that on this one that's being modified. And then they're showing their 5 broad classes of modifications. These would be post-translation modifications. Okay, acetylation, methylation, ubiquitination, sumoylation, and phosphorylation. So, let's start these. I'm actually going to start with one that is different. I'm not going to start with acetylation, okay? Well, let me get a different color. We're going to start with ubiquitination. Sometimes it's also called ubiquitinylation because of the fact that, what are we adding when we do ubiquitination or ubiquitinylation? What does it sound an awful lot like? What? Ubiquitin. And what is ubiquitin? What kind of biomolecule is it? Protein. It is. It is actually a protein. What does ubiquitin do? There are 2 main functions of ubiquitin. One is it's involved in the activation of proteases, okay, and which ultimately helps do what? Degrades what? The proteins, okay? It'll kill off the protein, so to speak. That is not what it's doing here. This is number 2, okay? And so this has to do with transcription. And so, but that's why I pointed out. Ubiquitin gets its name because it's ubiquitous, which maybe I was done for too long. I assume that people knew what ubiquitous meant. If you real perhaps you know, what does ubiquitous mean? It's everywhere. It's everywhere. It's everywhere. Yes. It's ironically it wasn't in cancer. They didn't know what ubiquitous meant. Bada bing. All right. 76 amino acids. Not a huge protein. It's 8.5 kilodaltons. Those are relatively small. Usually, it's about protein degradation. When proteins start to get ubiquitinated, and it starts to the higher degree of ubiquitination that you get, the more likely that the proteases are going to, the ubiquitin-activated proteases are going to recognize, okay, something's wrong with this protein, we need to chew it up and get rid of it. That's not what we're doing here with histones. So you don't want to just chew up the histone and the DNA and everything around there and get rid of it. But it does actually help regulate transcription, because what it does is it's going to help decrease the affinity for the histone for the DNA. So if it's decreasing the affinity for the histone for DNA, is that going to increase transcription or decrease transcription?
Increase.
It's going to increase it. Anytime that you're relaxing the DNA from the histones, you're going to increase transcription. Okay. And so I'm just going to go back real quick. Okay. Sumoylation is very similar. Sumo, I don't remember what sumo stands for. And sometimes they call the human version of sumo humo. Sometimes they call them mouse. But I mean, they're all homologs of each other. It's a little bit larger, but it's very similar to ubiquitin. It's just it gets added on, and it also helps initiate the enzyme cascade in order for transcription to occur. I'm not even going into a lot of detail, other than for you to recognize sumo, and it's called sumoylation because the YL once again means it's being added to something. This is where I want you to really start to think about it, because from organic chemistry, you should be able to understand this. Oh, just real quick, I want to show you. This is acetylation. Okay, let's go back to heated. All right, so I took this picture of an acyl group. Technically, the one that's shown here is called an acyl group. I took it off of Wikipedia. Okay. So the lysine would be getting attached right there. So let's say that we have our histone. 1, 2, 3, 4. That's why it was not made for lefties. 1, 2, 3, 4. What does a lysine amine often look like?
Proteinated.
It's protonated, okay? And if it's protonated, what else is it going to look like? What's missing?
Positively charged.
It's positively charged. Okay. So whenever it gets acetylated, once again, that's the actual histone. We have 1, 2, 3, 4. There's a nitrogen, carbonyl, and that's a methyl group. This is reversible. These modifications are reversible. What happens to the charge on the lysine? What's the charge now on that lysine?
Not positive.
It's not positive. Is it negative?
I guess it would depend on pH.
I mean, no. Is it negative?
No, this ain't.
No, it's not. Okay. So it's hard to pull the hydrogen off of that to make it negatively charged. So that's an oxygen. Sorry, it just didn't come out very well. So now it's neutral. So this is the acetylated. This is acetylated. This is deacetylated. Okay. Which one of these is going to tend to bind DNA for the histone? Right.
The one on the bottom.
Which one's going to bind? So you're saying that the one here is going to bind DNA more tightly?
Oh, no. Top one.
The top one. And it's going to bind DNA more tightly because it is positively charged. So this one tends to bind DNA more tightly. Oh, whoops, I thought I was writing. Well, not NA, DNA more tightly. So the KD would be smaller. This one, we're going to bind DNA more loosely. So the KD would be larger. Okay. So the enzyme that does this, what do you suppose it's called, that adds the acetyl group onto it?
Transferase.
It is a transferase. What specific?
Acetyltransferase.
That's it. That's all one word, by the way. I just don't have enough room. Transferase. So this one technically is a histone acetyltransferase, which sometimes I abbreviate as ch8 or HAT. And not only that, but there are specific histone acetyltransferases that are specific for certain histone proteins at certain locations that are highly, highly specific. Okay. What enzyme do you suppose is going to take it off? It's not the same enzyme, by the way. It's one way that you can control like whether or not the histone is acetylated or deacetylated. What kind of enzyme are you supposed to deacetylate something?
Is there only one type of enzyme that does that?
Yeah, I mean, this is one name that do. If you're going to deacetylate, you call the enzyme a what? It's a deacetylase. I mean, technically, under those 6 categories, it's going to be a very specific one. But the common name for those in that family is called deacetylases, and these, in particular, would be histone deacetylases, and they are also very, very specific. I know that there's at least 8. The reason why I know that is because of one of the people in my own lab. She worked on HDAC8, which is histone deacetylase number 8. And so I know there's at least 8 of them in humans. So, but yeah, these would be the histone deacetylases. So I wanted to point that out because I'm going to actually go forward. Okay. And just briefly, I wanted to go through the last 2 together. And you can just start thinking about it. This is methylation and phosphorylation. For methylation, many times we talk about a lysine getting methylated. Not only can lysine become methylated on that nitrogen, but the degree of methylation is also important because you can be monomethylated, dimethylated, and trimethylated. Okay? And so some people call it the methyl. And especially in cell biology, they like their owns, and some of them don't. Some of them call it transcriptome because of almost like language of the histones. But so the degree of methylation, because if it's trimethylated, it's a permanent positive charge on that because it's got 3 methyls on it. Now, on the other end, on the other side, we have the serine, which what's the function become a serine? OH, which is an alcohol, and it can become phosphorylated by a phosphotransferase or kinase. Okay. And so whenever it becomes phosphorylated, I'm just going to look here at the draw a serine. CH2. And it becomes, and once again, it's going to be reversible. What do you suppose is going to happen to that serine? What's missing? And the phosphoserine down below.
Negative charge?
Yes. Not only, but the way I have it drawn, how many negative charges? 2, and the minus here and the minus there, because it's depending on pH and pA. But yeah, so whenever, and once again, it can be a phosphatase can take it off, a kinase or a phosphorylase can add it on, depending upon where the phosphate came from. What is supposed to happen to DNA binding whenever it becomes when the serine becomes phosphorylated? Is it going to increase the ability for DNA to bind or decrease the ability for DNA to bind? Someone said it? Decrease, because it's negatively charged. So will that increase or decrease transcription? It will increase transcription. Okay. So that's just to show you the different types of modifications that can occur and the kind of rationale and theory logic that you have to start to think about. That's what they're trying to show here. That's one of the reasons why I liked this little picture. They're showing this one specifically for histone H3 K18, meaning lysine number 18, And see, whenever it gets deacetylated by histone deacetylase, it's got a positive charge. Whenever the histone acetyltransferase adds the acetyl group, it's now neutral. That's one of the reasons why I liked that figure.